Thank you very, very much, Uta, for joining us today in the Center of Desert Agriculture. I'm very, very happy, very pleased that you could come and talk to us about your work. Uta, she spent most of her career using genetics, molecular biology, and cell biology to understand the symbiosis between plants and mycorrhiza. She started her career back in Germany, where she did I think it was a Master's of Science with Jeff Dangel in Cologne in Germany. I think that was back in the midst of time when Arabidopsis was being established as a model plant system. And then Uta, she moved on to do a postdoc with Stephen Briggs. And this was, I think, judging from your publication record, I think this is when you started working on symbiosis. And you published a PNAS paper in 2002 on phosphate transport, which has been highly cited. And then you kind of switched sides and went from academia and worked in Syngenta for 3 years. And then you went back into academia, and Uta joined the University of Geneva as a junior group leader, and then moved to the University of Lausanne in 2006. And then in 2012, Uta took up her present position in the University of Cambridge, where she became a full professor in 2019. Now, before the seminar, I was debating in my mind, when was the first time I met Uta? And I'm not quite sure, so maybe you can help me with that. But anyway, my earliest memory I have is of a molecular plant microbe interactions meeting in Cancun in Mexico in 2004. And we all arrived, or many of us arrived 1 or 2 days before the meeting, but the day before the meeting, it was cancelled because of a hurricane that was approaching. And I wanted to stay in the hotel and experience this amazing force of nature, but Uta probably saved my life, and along with a few colleagues, they dragged me away from the hotel, and we got onto one of the last buses that was evacuating all of us to Mexico City. And that's a long bus ride. So after spending half a day in that bus, we got off halfway there and went and explored some beautiful Mayan temples. And then we went to San Cristobal de las Casas. And that was with Giles Albright, René van der Hoorn, and Martin Paniskis. So we had our own little MPMI meeting. And of course, I learned a lot about symbiosis on that trip. But of course, the field has moved on, and I'm sure there's a lot more to learn. And that's why I'm very much looking forward to your seminar today, Uta.
Wonderful. Thank you very much, Brande. And it's a great pleasure to see a number of known faces here on the screen. So thank you very much for joining, Mark. Lovely to see you again. And you can imagine, I mean, having gone through that rather extraordinary circumstance in Mexico means that the relationship between Brande, René van der Hoorn, Martin Paniskis, Giles, and me has an additional adventurous flavor that we probably will keep cultivating and as long as we can. Okay, I start now sharing my screen and hope it all will work out well. So you should be able to see the screen, the title, and my cursor. Thank you.
Yeah.
Okay, right. Wonderful. So let's get started. So here on the first slide, I'm trying to introduce to you the system that we're working with. We're looking at a plant root that lives in an ongoing arbuscular mycorrhizal symbiosis with soil-borne fungi. So you can see the root here, and you can see the fungal hyphae, the fungal spores. Perhaps you can also make out these little fungal structures, globular structures inside the root. So the outcome of that association is mutually beneficial, where the plant nourishes the fungus with organic carbon, The fungus is a fatty acid auxotroph, so it relies on the supply of fatty acids by the plant to complete its asexual life cycle. At the same time, the fungus quite dramatically, so significantly increases the surface area for mineral acquisition and provides a proportion of the mineral that it takes up to the host plant. So it indeed really increases significantly the surface area, the overall surface area for mineral acquisition. So the benefit the plants derive from this interaction can be as pronounced as shown here, where we're looking at a lateral root mutant of maize. So this particular mutant lacks all of the lateral roots, but still has the main crown roots, but is severely incapacitated in the exploration of the soil substratum as compared to the wild type. And when we grow this mutant in sand with reduced nutrient availability, we see that the development of this mutant is delayed. Eventually it ceases, it dies off, while the wild type is doing well under these conditions. However, the mutant, when supplied with one of these Arbuscular mycorrhizal fungi, can actually survive very well under these conditions. Wild type growth is restored, and that very nicely illustrates how the missing lateral roots can be functionally, and the emphasis here is really on functionally, replaced by the fungal mycelium. So this is what this kind of fungi have provided as a service to plants from the dawn of plant life on land. So the Arbuscular mycorrhizal symbiosis dates back to the early land plants, to the occurrence of the early land plants at approximately 450, 470 million years ago. It predates the emergence of roots, so most likely it has been this service of exploring the terrestrial environment that has allowed plants to make the transition from water to land. Roots evolved later. Plants split up into these different lineages in the course of the evolution of what we see as today's plant kingdom. And ancestral to the root evolution has been the mycorrhizal symbiosis. So we see still today mycorrhizal competence in the vast majority of the extant land plants in more than 80% of all existing land plants. So that implies actually that the minority of plants that do not engage in the interaction have secondarily lost the ability to interact with these fungi. Amongst these, we find the members of the Brassicaceae. So Arabidopsis is a non-host, cannot be used for the study of Arbuscular mycorrhizal symbiosis. So just a few words on how the symbiosis develops. It starts off by the spores of the fungi residing in the soil and germinating upon the right moisture and temperature. The germ tube that would grow eventually gets into the vicinity of a host root and perceives host-released factors. Foremost here to be mentioned are the strigolactones, the plant hormone, strigolactone, that is released in considerable amounts into the rhizosphere, perceived by the fungi. It activates the fungus for the symbiosis and also navigates it towards the root. At the same time, the fungus is releasing a cocktail of compounds, best known the chitinaceous molecules that now are perceived by the plant. And it is this pre-symbiotic molecular crosstalk that enables reciprocal recognition and also reprograms both partners for the anticipated physical alliance. And only when it occurs accurately does the fungus then differentiate a so-called hyphopodium on the root surface. This is the first physical interaction between the two organisms, and it is here where the fungus now penetrates into the root tissue. It colonizes the outer root cell layers, the rhizodermis and the cortex. It does not breach the endodermis, and it does not grow into the meristematic tissue. So as it proliferates in the inner cortex, in the intercellular space, it also starts to produce these so-called arbuscles, little tree-like structures that are produced, in fact, by an invagination of the fungus into the cortex cell, invaginating the plasma membrane of the cortex cells, and by reiterative dichotomous branching, eventually building these tree-like haustoria feeding structures, fungal feeding structures that eventually entirely fill out the cortex cell. As you can see here on the left-hand side micrograph, you see the plant cell wall, and you can see in green here the fungal canopy of an arbuscle. So obviously, the hosting of such a fungal structure is accompanied by monumental changes in plant cell architecture. And foremost to be mentioned here is the enormous de novo production of plasma membrane material to envelope this growing arbuscle, fungal arbuscle. So the arbuscles are the heart of the symbiosis, are the heart of mutualism of this interaction, because it is here where the exchange of minerals for carbon occurs. So despite their importance, and despite what seems to be a major investment of both organisms to produce these arbusculated cells, the arbuscles, they have a relatively short lifespan and collapse within the course of just a few days. The fungus leaves the cortex cell, and the host cell architecture is completely restored to the state prior arbuscle hosting. So we have a highly dynamic process of arbuscle development and collapse within the course of just a few days, and this must be clearly the result of a very fine-tuned, likely complex exchange of signals that governs these highly compatible type of interaction. And if the fungus is then well nourished by the plant with fatty acids, with carbon, it would form these vesicles inside the root, so these are lipid storage structures, and would also grow out into the soil, forms this far-reaching mycelium, which is involved in nutrient acquisition, and is also the site of asexual spore development, from where the cycle can start again. So my lab overall is interested in the elucidation of plant-fungal communication. We work almost entirely on the plant side because the fungus is very intractable, it's genetically intractable. There's no transformation protocol, there's no genetics. So we focus on the plant side and would like to understand how communication, how signaling to the fungus and perception and integration of fungal signals by the plant works. And over the course of the years, we have settled on two stages predominantly with our studies: the pre-symbiotic molecular crosstalk and the processes that underpin this fascinating arbuscle development. So I've prepared two stories for you today to give you a flavor of what we are very much involved with at the moment, and I'm looking forward to hearing your feedback about it. So first of all, looking at pre-symbiotic, the pre-symbiotic stage a few years ago, we identified a virus mutant that ticked all the boxes for being impaired in perception of AM fungi, Arbuscular mycorrhizal fungi. So what you see here on the micrographs on the left-hand side is a typical image that you obtain of a colonized wild-type root stained with trypan blue that highlights in dark blue all the fungal structures inside the root. But we completely failed to see anything like that in this particular mutant. We could make out the fungus on the root surface, but we did not find any evidence for physical engagement with this mutant root. At the same time, when we then looked at transcriptional responses of the mutant and wild-type 2 fungal exudates, we found that the mutant entirely failed to mount a diagnostic transcriptional response. So together, these two phenotypes strongly suggested that this mutant was indeed perturbed in perceiving Arbuscular mycorrhizal fungi, the strongest phenotype known so far for any plant mutants. So we eventually cloned the gene and were surprised to find that it is actually a very well-known gene, the so-called dwarf 14-like in rice, also called Carykin insensitive number 2 in Arabidopsis, and it corresponds to the Carykin receptor. So Carykin is a compound that you would find in bush fire, in smoke of bush fire. So it is a botanolyte that many fire-following plants require for the initiation of seed germination. And the receptor that mediates perception of this Carykin leading to seed germination is this gene, or is this protein that we tapped on here with our Symbiosis mutant. Now, quite important to note also is that D14-like is in fact the evolutionarily older paralog to the strigolactone receptor D14. It's a horrible nomenclature, so I hope I keep you on board with the different, very confusing names. So the strigolactone receptor indeed evolved much later in the course of plant evolution with the more and more complex vasculature and above ground architecture of plants. And if you look at the two chemical formula here, then you might think that these are actually rather different. Well, they are not with regard to the botanolyte ring. So the D ring in strigolactone and the botanolyte ring here on the right-hand side in the case of Carykin is in fact what is bound by these receptors, is what enters into the binding pocket to then kick off signaling. So now, having this identified this D14-like gene as being so centrally important for Arbuscular mycorrhizal development, what do we actually know about Carbicene signaling? Well, that has historically been largely guided by numerous efforts in the area of strigolactone signaling, where we have learned that the binding of strigolactone by the D14 receptor leads to hydrolysis of the strigolactone molecule. The D ring covalently binds to the receptor, which introduces a conformational change that then allows the recruitment of the F-box protein D3 in rice, more auxiliary buds in Arabidopsis, and leading to the ubiquitination of a negative regulator that becomes degraded by the 26S proteasome machinery, and thereby liberating, de-repressing genetic programs that underpin, for example, shoot architecture that I believe we're all familiar with as one of the major developmental processes that are regulated by strigolactone. Now, in the case of Carykin, we're lacking some biochemical information still, but basically the picture is largely similar. The binding of Carykin by the D14-like receptor leads to the recruitment of the very same F-box protein D3, ubiquitination of a related but not the same negative regulator SMARCS1, which sends it for degradation and enabling the de-repression of developmental programs such as seed germination. So this is what we have learned over years from numerous labs, largely in Arabidopsis. So can we actually extrapolate what we've learned here to rice and to Symbiosis? That was the first thing that we addressed. And we turned our attention first to developmental processes that are controlled by the D14-like /Chi2 alpha-beta hydrolase receptors. So in the first place, when we look here on the left-hand side at Arabidopsis, we tried to ask the question, if we introduce the rice gene into the Arabidopsis mutant background, can we actually complement the developmental phenotype, in this case, hypocotyl length? Because when we mutate the Carykin receptor in Arabidopsis, we see that the hypocotyl length is significantly enlarged. Introducing now the rice coding region under the 35S promoter into that Arabidopsis mutant restored the shortness of the hypocotyl back to wild type length, so illustrating that or confirming that the rice gene functions equivalent to the Arabidopsis gene in the Arabidopsis background. We then turned this question around and looked at the rice mutant. Also here we find now this is a mesocotyl, because now we are in monocots, that removal of a functional Carykin receptor led to an enlarged or longer mesocotyl relative to wild type, and reintroduction of either the rice gene or now introduction of the Arabidopsis gene under the native rice promoter equally restored the shortness of the rice mesocotyl equivalent to the wild type. So that means actually that the Arabidopsis Chi2 and the rice D14-like are functionally comparable or equivalent when it comes to the control of epicotyl elongation. So obviously these are all stably transformed lines. So we took now these rice lines and asked the question, how about complementation of the symbiosis phenotype? Here we have the controls. We have dark blue, the fungal structures, showing the intense colonization of wild-type roots. If we look at the D14-like mutant transformed with the empty vector control, we see no colonization coming in now with the complementation of the mutant by either the rice coding region or the Arabidopsis coding region. Under the native rice promoter, we see now a surprising difference in phenotype, namely that the rice coding region perfectly well complemented the rice symbiosis mutant phenotype, but the Arabidopsis gene failed to do that, and failed to do that in 7 independent transformants, despite the fact that it's exactly in these lines where we also saw developmental complementation. So such differential complementation could arise by a number of different scenarios. We're a bit scratching our head. We don't have an answer quite yet, but we're tossing a number of ideas. So it could be that the rice and the Arabidopsis receptor have simply different sensitivity. We need to note that in the mesocotyl/hypocotyl or root, we are in a different tissue, and a different tissue can have different cellular contexts. We might be dealing with different ligands, different affinities, and that could underpin different conformational changes and thereby lead perhaps to a different composition of the receptor complex, and depending on composition driving signaling into either the symbiosis or the developmental pathway. We don't know. We have a set of chimeric Arabidopsis rice proteins generated. We just received the transgenic lines, so I hope that we, within the next 2-3 months, will have an answer to some of these questions. So the next obvious step was also to verify what is the requirement of the other known signaling components of the D14-like pathway for symbiosis. And Jong-Min Choi, a former postdoc in the lab, had and in detail studied this question. So we would have expected that if we indeed deal with the same type of components, that we would reproduce the D14-like phenotype by mutating the F-box protein D3. And if SMUX1 would also operate as a negative regulator in symbiosis, we should see an increased colonization. And that is exactly what Jong-Min found, and this is what is shown here. This is a little bit of a busy slide, a typical slide. We're looking at total fungal colonization of the root, hyphopodia, intravadical hyphae, or arbuscules. all of these different structures give us a qualitative feel for how to interpret the interaction. And what we see immediately here is that the D3 mutant reproduces the very strong D14-like phenotype, whereas turning now to SMUX1, we monitor a significant increase in overall colonization as much as these individual structures relative to the wild type. Please note that we have a fairly limited colonization in the wild type, so that was on purpose. Sampled at a very early time point where we actually able to resolve increased colonization, as Jongmin monitored, in fact, here. So that was very encouraging for actually confirming similar roles of D3 and SMUX1 in D14-like signaling as we knew from the developmental field. Jongmin then also addressed whether or not SMUX1 is downstream of D3 by bringing the SMUX1 mutant into the D3 mutant background, and could confirm that this would restore the wild type phenotype, so wild type colonization, whereas if in siblings where we have at least one functional SMUX1 left, we fail to restore wild type colonization. So together that means that SMUX1 is a negative regulator of symbiosis, and this epistatic analysis confirmed that it functions downstream of D3. So that really means that the point of activating D14-like signaling is to get rid of SMUX1, to take the foot off the brake, to then de-repress symbiosis programs. So if that hypothesis is true, then we should actually see an accumulation of SMUX1 in the D14-like and in the D3 mutant backgrounds. And that is exactly what Jongmin found. Here looking at GFP-tagged SMUX1 and using anti-GFP antibody, we can detect a very strong band for SMUX1 in D14-like and in the D3 mutant background, but not in the wild type. And confirming that when D14-like signaling is not activated, that SMUX1 is stabilized. What we were a little bit surprised, we had thought that we would see an increased accumulation, so some band in the wild type and then more of the protein in the mutant, but actually we've completely failed to see any evidence for SMUX1 in the wild type, which we believe is noteworthy because it seems that this D14-like, it seems to indicate that the D14-like signaling pathway is constitutively on, perhaps, in the wild type. So what is SMUX1 now actually suppressing? And that could be relevant for symbiosis that underpins that strong phenotype. So Jongmin went ahead and did an Rna-Seq experiment, including all of the different genotypes that we had characterized in this context. I'm showing here only the summary of the data for the SMUX1 single and D3 double mutant. So she found that overall there were approximately 400 genes that now were upregulated as a consequence of genetic removal of SMUX1. She overlaid these significantly upregulated genes with genes that we know are regulated in the wild type in response to symbiosis, and also overlaid this with a set of genes that are conserved in the genomes of plant species that engage in arbuscular mycorrhizal symbiosis and are absent in genomes of plants that no longer engage with the fungus. So there is a considerable overlap in all of these different comparisons, and I'm not going to dwell much more on that because further functional analyses are ongoing. But what Jongmin noticed as an interesting signature was that there was an overrepresentation of genes that we are familiar with from the strigolacton biosynthetic pathway. And Jean-Min then took a careful look at the entire biosynthetic pathway of strigolactone, starting from isoprenoid biosynthesis going into the carotenoids, and then the committed pathway into strigolactone biosynthesis, looking across the different genotypes as shown here. So we have SMUX1 in the DOM gene background, we have D3 in the Shiokari background, then we have the double mutant and the sibling with a wild type copy of SMUX1. So you see a pattern that is consistent across the entire biosynthetic pathway. Namely that in the SMUX1 mutants, we see a transcriptional induction of the respective biosynthetic gene. So then Jean-Min collaborated with Haro Baumeister to actually measure strigolactone and see if that transcriptional signature would be translated into a physiological response. And as a matter of fact, that was the case, looking at 4-deoxystrigol and epistrigol in wild type D3, now the strigolactone receptor mutant, and then the D3 SMUX1 double mutant. And I think we all agree that indeed there is a significantly higher production of strigolactones in the SMUX1 D3 double mutant. So that indicated an unexpected crosstalk between the D14-like signaling and the strigolactone biosynthesis. But then Jean-Min also found that there was another overrepresentation of very well-known gene to us in the plant beneficial microbe arena, namely genes which belong to the so-called common symbiosis signaling pathway, a pathway that is in legumes equally required for nodulation as much as for Arbuscular mycorrhizal symbiosis, and that in the non-legumes is equally required for interaction with Arbuscular mycorrhizal fungi. So here we all of a sudden find a whole signature of several of these common symbiosis signaling components being transcriptionally regulated in a D14-like dependent fashion. And what we believe is the case is that the simultaneous regulation of these genes may eventually explain, respectively the absence thereof, may explain the strong phenotype in the D14-like and in the D3 mutant backgrounds. So what I would like to summarize here and draw your attention with, something we are really excited about as an outcome of this study, it seems that the activation of the D14-like signaling pathway coordinates the fungal attraction through strigolactone. Remember, I told you that in the introduction, that strigolactone is released by the plant into the rhizosphere to condition the fungus, activate the fungus for symbiosis. And the D14-like signaling pathway seems to be required for that. At the same time, it is required to open the doors for the fungus. So it coordinates the simultaneous calling for the fungus, attracting the fungus to the root, and opening the doors for the fungus to then colonize. In a more sort of mechanistic way, what I would like to summarize here is that when we see SMUX1 stabilization, possibly under non-favorable conditions for the symbiosis, we believe that this stabilization suppresses essential transcription factors that regulate strigolactone biosynthesis, that regulate the symbiosis genetic program. Under these conditions, nothing of that is happening because the transcription factor is repressed and we don't get strigolactone biosynthesis and Arbuscular mycorrhizal symbiosis. However, under favorable conditions, and one very plausible scenario is, let's say, low phosphate availability is what is long time known as a favorable condition for plants to engage in these symbiosis. So let's pick up on that idea that under low phosphate nutrient availability, there is an endogenous ligand this is hypothetical, of course, an endogenous ligand that is bound by D14-like, kicks off the D14-like signaling pathway that leads to the degradation of SMAX1, liberating the transcription factors that now transcriptionally activate the strigolactone biosynthesis, leading to increased strigolactone secretion into the rhizosphere and activation of the fungus. At the same time, symbiosis genes are turned on as much encoding components in the nucleus or on the plasma membrane, enabling symbiosis to develop. Of course, what we are really hot on elucidating is what are these transcription factors. So let's switch gears from the pre-symbiotic molecular crosstalk to this highly dynamic stage of arbuscular development. So how to study this, how to zoom into this arbusculated cell type. It is incredibly challenging, and with this micrograph here, I'm trying to just illustrate what makes it so particularly challenging. We're looking here at a cross-section through a rice root that is colonized, where we see quite a number of arbuscles highlighted here in blue because we have the symbiosis-specific phosphate transporter from rice PT11. Promoter driving the GUS gene. So all of these blue cells indicate arbuscles because that promoter is only active as the fungus starts to invaginate the cell and produces these arbuscles. So the first thing we note is that we actually have only a proportion, of course, of the root cells that are simultaneously hosting an arbuscle. As I told you in the introduction, arbuscles are transient and form also asynchronously along, let's say, the cell files that are consecutively producing where the fungus consecutively invaginates and produces these arbuscles. I should also mention the whole colonization process is not synchronous at all. The fungus can grow out into the root rhizosphere again, so into the soil, and can re-infect. So it is, once you have the first initial root colonization stages passed, we actually see all of the stages present at the same time. So that poses another complication when you actually want to study specific stages of arbuscle development. And then because it's highly dynamic, it would be desirable to include live imaging to follow the dynamics, capture the dynamics of arbuscle development, but visual access in the inner cortex by confocal microscopy is becoming increasingly feasible, but has been a big technical challenge as well. So what was the approach that we took a little while ago was to try to identify proteins that have a regulatory role in these arbusculated cells, plant proteins once again, that must participate in that regulation of this, in the orchestration of the signals that underpin the dynamics of this highly compatible cell state. And we focused on receptor-like kinases as the best guess for type of proteins that would underpin such mechanisms. But how to get to these? Our approach was to turn to maize. Why to maize? Because maize has this nice attribute that as the root tissue becomes arbusculated, it accumulates a yellow pigment that allows us to actually enrich for arbusculated tissue. From here, we then did a membrane proteome analysis of mycorrhizal versus non-mycorrhizal maize. We then overlaid the membrane proteome with laser capture microdissected transcriptome specifically for the arbusculated cell, so that from the large list of proteins occurring in mycorrhizal maize roots, we could zoom into those that would actually occur in the, would have a high chance to occur in the arbusculated cells, based on the matching transcriptome profile. And in the resulting gene list, we would then look for the receptor-like kinases, and I should say gene list / protein list. And among the receptor-like kinases, we then identified ARK. ARK stands for Arbuscular Receptor Kinase. It is a little bit of an exotic receptor kinase because it has a very short extracellular domain of only 22 amino acids, followed by a transmembrane spanning domain and a functional cytoplasmic kinase domain. What is interesting about this particular Arbuscular receptor kinase, ARK for short, is that it's one of those genes that you find strictly present in all plant species that are able to engage with Arbuscular mycorrhizal fungi and strictly absent in plant species that cannot. And it's so extreme in the case of ARK that, for example, Markantia polymorpha, which is a non-host, does not have the gene. Markantia palliacea, which is mycorrhizal, has the gene. So now, is it cell type specifically expressed? Does ARK occur in arbusculated cells? So we first looked at a transcriptional reporter and can confirm by looking at the promoter, ARK promoter driving Gus, that we see Gus staining only in the arbusculated cells. How about protein accumulation? Is it in the periarbuscular membrane or is it in the peripheral plasma membrane that surrounds the cell? So from confocal microscopy, it looked very promising that ARK would reside in the periarbuscular membrane. We're looking here at translational fusion with RFP driven by the native promoter. And we compared this to, if you wish, our reference protein on the periarbuscular membrane. Once again, the Symbiosis specific phosphate transporter from rice PT11, here fused to GFP under the native promoter. So this is the one and only phosphate transporter mediating symbiotic phosphate uptake, and it resides in the periarbuscular membrane surrounding the arbuscular fine branches. So this was encouraging for ARK1 to eventually reside on the periarbuscular membrane. We then went ahead and to confirm the subcellular localization by immunogold labeling. And you can see that indeed ARK resides on the periarbuscular membrane. This is the cross-section through a fungal hypha inside a cortex cell. Here the magnification. And so indeed ARK1 is localized on the periarbuscular membrane. So can we now resolve when during arbuscular lifespan ARK comes up on the periarbuscular membrane? And once again, we now first turn to our reference protein, if you wish, to PT11, because it was about at the same time that Ronald Ross, a former postdoc in the lab, now a university research fellow at Oxford University, she established multiphoton confocal microscopy, live imaging in the lab. So all of these images I'm going to show you are non-invasive protocols where we just stick live roots under the multiphoton confocal microscope. And she once again first turned to, as I said, to PT11, to the phosphate transporter, to try to capture different stages of arbuscle development. And what you see here is, a little bit assisting with this line, is a young arbuscle where we see PT11 surprisingly actually occurring first in such foci opposite bifurcating fungal tips. So that was a new observation. As the arbuscles then mature, we met an image of distribution that we're more familiar with from the literature, a more even distribution along the arbuscular branches, so on the periarbuscular membrane surrounding the arbuscular branches. And then when we come to a senescing arbuscle, we now see evidence of asynchronous collapse and asynchronous removal of PT11 from the plasma membrane. So how about ARC now in this context? So a present senior grad student in the lab produced a rice line that co-expressed PT11 GFP under the native PT11 promoter and ARC RFP under the native ARC promoter. And looking at young, mature, and senescing arbuscules and in the same cell at the two different proteins, she found that in young arbuscules, she only found evidence for PT11 GFP signal. And she made the effort to actually draw the exact structure that she saw in the microscope. Whereas at this young stage, we can hardly detect any RFP signal. If we now come to the mature arbuscular stage, then we see both of the proteins present in the periarbuscular membrane surrounding the fine branches. But then another surprise was waiting for us when we came to senescing arbuscules, where we now do not see at this stage any GFP PT11 signal anymore, but a very strong signal for the receptor kinase ARC1 RFP. So that points to really differential dynamics of periarbuscular membrane proteins, with PT11 being present more at early stages, and then ARC1 overlapping at the mature stage, but being present also at the senescing stage. So what does ARC1 actually do? What do we know about the role of ARC1? Well, we had two TOS17 retrotransposon insertion lines that led to null alleles of ARC1, and comparing in the first place the fungal structures inside ARC mutant roots with that of wild type, we were a bit disappointed to see that there was no evidence for ARC1 being required for arbuscle development. I mean, ultimately, we saw expression of ARC1 in this particular cell types that host the arbuscle, but yet removal of functional ARC1 had absolutely no impact on arbuscle development. However, when we then looked at colonization at 3 weeks and 6 weeks post-inoculation, we initially do not see any difference in the colonization of wild type and the two mutant alleles, but we do so at the later stages of infection, when we see a significantly reduced overall level of colonization, reduced arbuscle numbers, but also, and this is most striking, reduced vesicles. So this is a new phenotype relative to published work. So to have actually a stage post-arbuscle development that seems to impact fungal fitness is a sort of pointing towards a new stage that is under plant control. So we interpret this phenotype as a compromise in fungal fitness. And this can be experimentally addressed by growing plants in a so-called nurse system. So we have a tester plant and which grows together with a donor plant in a small little petri dish. The donor plant is planted first, inoculated, allowed to be fully colonized, and then we add the tester plant. So now the fungus is super vigorous because it's nourished by that donor plant. And does that make a difference for the mutant phenotype? So here you have first the three controls, donor and tester of the same genotype. We reproduce once again the same significant reduction in the mutant relative to the wild type. But what happens now when we grow ARC1 mutant alleles with the wild type? Well, we could fully complement the mutant phenotype, and that confirms that ARC function is somewhat underpinning fungal vigor. And what springs to mind is that this might have something to do with fatty acid nourishment of the fungus. So in order to address that, Hector Montero, a former PhD student in the lab, established a new dye, lipid staining procedure in the lab. So what we are looking at here is wild type roots colonized by our Arbuscular mycorrhizal fungus. WGA, wheat agglutinin, specifically stains chitin, so you would only see the fungus here. This is the new lipophilic dye, which unspecifically stains all of the lipids, and you can already see, let's say in this cell here, we have a developing arbuscule. We see that this dye outlines the fungal hyphae. So in this case, the lipids are inside the fungus. However, when we turn to these cells here, we see a very even distribution of the lipids now in the host cells. And when we overlay the two images, then we can see that in the case of these mature arbuscules, in the cells that host mature arbuscules, these cells, host cells, crank up the lipid biosynthesis, and to the effect that we can actually see the accumulating lipids in these cells. What you can't easily see from this picture, I apologize for that, is that actually mock cells, so non-colonized cells, don't show any lipid signal. So when we now turn to our ARC mutants, then we find that in the case of the arbusculated cells, remember, we still get arbuscules in the ARC mutants, which look morphologically normal, but we've completely failed to see that accumulation of lipids in the host cell that now harbors an arbuscule. So that would furthermore be consistent with ARC1 signaling modulating fatty acid accumulation. We don't know how. We are actively working on this, and we would love to elucidate how post-arbuscule development fungal fitness is ultimately modulated by the plant. So this brings me to the summary slide of that section. So what I think is really exciting, and I would like to draw your attention to that, is that we now seem to start to be able to actually pinpoint different stages of arbuscule development being relevant for different aspects of that bidirectional nutrient exchange that underpins mutualism. We see PT11 coming up only in the expanding young arbuscule. So it seems like the symbiotic phosphate uptake is linked to the process of expansion and not to an arbuscule that sits there and where PT11 is pumping phosphate delivered by the fungus into the plant. We have no evidence of any static state of an arbuscule from our confocal analysis. Likewise, when we now look at ARC and the phenotype, the localization and the phenotype, together suggest that fungal fitness is, at least to a certain extent, linked to the mature state of arbuscules leading into collapse. So that, I think, changes a little bit our understanding of how that bidirectional nutrient exchange is ultimately occurring on a cellular basis. So this now is my last slide, the take-home messages. So what I hope I was able to convince you about here today is that activation of the D14-like signaling pathway enables arbuscular mycorrhizal symbiosis by removal of SMAX1. I introduced you to a new dye and fatty acid dye or lipid dye, which suggests that fatty acid accumulation in arbusculated cells is ultimately modulated by a new receptor kinase, ARC1. And last, I hope I provide you some food for thought about arbuscule dynamics being relevant for the reciprocal nutritional benefit these two interacting partners derive from one another. And this is us. So I've been really blessed with a wonderful set of people in the lab. I would like to highlight Jongmin Choi here, who has done this beautiful work on SMAX1. She is, as of this academic year, advanced to junior group leader here at the Crop Science Center. So she will continue to work on arbuscular mycorrhizal symbiosis in rice, focusing on nutritional signaling in this beneficial interaction. And I have a bunch of 8 PhD students here that are an absolute gem to work with. One of the really fantastic sides of being in Cambridge is you get really, really fantastic students, many of which you know from college from day one. And we've had phenomenal collaboration. Emma Wallington here at NIAP does all our rice transformation. Giles is a physical neighbor. He's the head of the Crop Science Center here. We work a lot together. And Tak Lee and his group has helped with the RNA-seq analysis, Haro Baumeister strolactone analysis, and we got the SMAX1 mutant from Jinyang and in Korea. I can't praise my alumni enough. Ronelle has established live imaging for us in rice, in mycorrhizal symbiosis. It's amazing what we are able to see now, and it's to her credit that we can. And Marco, Caroline, Enrico, and William are former PhD students and postdocs in the lab. We've been quite lucky with funding over the last years, and I stop here and I'm happy to take questions. Thank you.
Beautiful, beautiful seminar and very, very beautiful cell biology. Uta, thank you so much. Questions?
Thank you.
Salim.
Actually, students should start. I will.
Oh, yeah, true. Yes, no, we should. We're trying to implement that students and postdocs should ask the first question. You're right. Thanks for reminding me, Salim. So please, students, come forward. Ting Ting.
Hello, thank you for a nice talk. Yeah, so I have a question concerning to the traditional signaling pathway of symbiosis that we all know that is so classic, and you said that it's overrepresented. So for me, especially for CKMK, it's so crucial that if you mutate it and then nothing happens, and it's nicely followed with the receptors, and you know. I feel if you could explain it a bit further. So
yeah, sure. Yeah, with pleasure. So what I mean overrepresented is just if you look at the overall list of genes that are differentially expressed, you see several of these common symbiosis genes all of a sudden being differentially expressed. So as if they were transcriptionally regulated in a SMAX1 dependent fashion. So if we knock out any of these common symbiosis genes in rice, we get a quite strong phenotype, but not as strong as D14-like and D3. So we still get hypopodia formation. Hypopodia look a little bit funky, but they are still hypopodia, and the fungus also can still get into the epidermal cell layer, and usually it's then blocked. So we sort of have a little bit the lab jargon to say common symbiosis genes, individual mutants have an impairment in cortex invasion, and that flavor holds true if you look actually at other plant species, other plant common symbiosis mutants as well. So, but D14-like and D3 is so much more strong. I mean, there's just no interaction whatsoever. So we could not really bring together let's say comparison of these two different phenotypic strengths without proposing. So it is in the absence of having yet data and any better idea. So what we are proposing is that by perhaps simultaneously suppressing a number of these different common symbiosis signaling pathway components, you arrive at that very strong D14-like phenotype. We hope to be able to form an answer to that soon, but this may turn out to not be entirely correct. But this is as good as it gets at the moment. What I can add is that we have some recent data that when we introduce an autoactive version of CCAM-K into the D14-like signaling, into the D14-like mutant background, we rescue colonization. But we do not rescue colonization if we just overexpress CCAM-K in D14-like. So you need the autoactive version to get that level of complementation. Yeah.
Okay, thank you. Thank you for the explanation.
Thank you.
Yeah.
Any more questions from students?
I don't buy it.
Then I will.
Salim, please.
There is a question in the chat, I think.
Oh, let's go to the chat then.
Leonardo Fabian.
Leonardo says, I have a technical question about the quantification of the symbiosis rate. In which way do you measure this value? What do you count? Is a standard measure or is it an internal one?
So this is a very good question, and we have a way that by which we count colonization that is practiced by many labs for many years, but it's not the only way. So you're totally right that one can think of different ways to try to express qualitatively as much as quantitatively what is happening. So if we just, and I'd like to just spend a moment on that because I think it's hugely important. It's a really great question, so thank you for that. When you have wild-type colonization, so this is, once again, this is dynamic. The fungus is approaching the root, is getting into the root, is proliferating into the root. But the whole root system is growing at the same time. So it really depends on how you inoculate, how much inoculum you present, on which type of, let's say, colonization kinetics you would see. So to have a uniform measure needs a little thinking. And what we do is what is called the intersect procedure. So basically, we collect representative root pieces from the root system. I mean, in the case of rice, maize, these are relatively large, so you don't need to stain the whole thing, but you basically chop off the root system, chop it into 1/2 inch, 1 centimeter large pieces, take representative samples, stain them, and then you mount them on a slide, the 10 pieces, and for each of the piece, you stop at random 10 places and monitor what fungal structure you see. So thereby you get 100 data points. And as we say, this is a representative set of root pieces for the root system, we express the colonization as percentage of total root length colonization. And then you break this down into total. And then for the symbiosis state, we start from hyphopodia, we have intravatical hyphae, we have arbuscles and vesicles. These are the 4 structures that give you an idea about where you're at with the symbiotic state. But we do also monitor extravatical hyphae, we do also monitor spores, and the holistic picture gives a very nice overview. Having said that, every data point is seeing 3 biological replicates within the experiment. So saying that you look at least 3 parallel plants, right? And then you reproduce the experiment as well. So it should be pretty robust. But the statistics that we do from within the same experiment, the different biological replicates, the different plants from within the same experiment, because an additional experiment will grow differently. And if you then do the stats across that, will give you a wrong, actually, picture eventually. So it's a little bit of a complex thought how to properly do phenotyping, quantitative phenotyping. And then, of course, qualitatively is where we use that WGA stain, where we then can zoom into morphological details. So that is no longer for quantification. That is then for qualitative assessment of the phenotype. Does that answer your question? Good.
He says thanks in the chat.
Yeah, I see it. Thank you.
Okay, Salim.
I think I'm allowed to ask now. I have actually many questions.
Shoot.
You work about, the D14 lichen rice is amazing. But as usual, you solved few questions and you raised many others.
Yes.
Now, you assume that there is a signal that is arising upon phosphate starvation, binding to the Carykin receptor and triggering strigolactone biosynthesis. And in principle, it is making the plant accessible to the mycorrhizal.
Exactly. Yeah.
Now, I mean, if I continue, you think that this compound is not present in Arabidopsis? I'm coming from the other part of the story.
Yeah, sure. I mean, the D14-like receptor is everywhere across the plant kingdom. Every plant has at least one gene that encodes this Carykin receptor, and we can trace it back to the charophytes. So there is a prototype even in the green algal ancestor of plants. So it is known to integrate a number of environmental cues. So Carykin, in the case of the fire-following plants, it does integrate Carykin signaling in Arabidopsis. So I would say the breakthrough several years ago in the lab of Stephen Smith in Australia was that you could break seed dormancy of Arabidopsis ecotypes that had a bit of dormancy by addition of Carykin. And that's how they were able to identify Ki2 first, and as Carykin insensitive, right? And so that means despite Arabidopsis not being a fire-following plant, there is still an effect of Carykin on seed germination. So let's say seed germination is one process, drought is another process. The very first report of this gene and the very first name of this gene was hyposensitive to light. So it's required also for photomorphogenesis. So we see, the more time passes, the more sort of jobs appear on the list of what this receptor does. Yeah, sorry.
But you are actually postulating the presence of more than one ligand, and I like it so much.
Yes. And
does Carykin trigger strigolactone biosynthesis in rice?
So Carykin, we did two things. We added Carykin to rice roots and saw what would be the effect on the symbiosis. 0. We added Carykin, so there's Carykin 1 and Carykin 2, which have been described as very active in Arabidopsis with some preferences, whatever. And we did Rna-Seq on rice roots treated with Carykin 1 and 2. There's not much happening. So I think that we need to be prepared for a number of ligands. We know in terms of synthetic ligands that there is indeed a relative of GR24. So when we add GR24, it's a racemic mix. So it has a number of slightly different racemic forms chemical forms in the mixture. And while D14 is the strigolactone receptor, D14-like can actually bind an enantiomer, which is called 5DS, and trigger D14-like signaling. So I think, and there's also evidence from the lab of Caroline Gutia, I think that we probably are confronted with different ligands, and different ligands perhaps in different tissue. Yeah.
And the connection between Ki2 and strigolactone biosynthesis probably only in mycorrhizal plants. In Arabidopsis, they are not connected. You know, you cannot trigger strigolactone biosynthesis by adding carykin.
Well, we can't do that either. No? So we can't. It's not that we add carykin. I mean, we haven't done that. I should say we haven't done that experiment. We haven't measured the effect of. We haven't. This I have to be careful. We haven't measured the effect of carykin treatment on strigolactone biosynthesis in rice. What we have is that genetic link. Yeah, and whether that exists in Arabidopsis, I don't know.
I have many other questions, but I think other people should also ask. Just one small question. You know, Michael Walter supposed many years ago that mycorrhizal and other small carotene-derived compounds are actually a kind of clock for the life of the Arbuscular.
Yes.
Do you have any indication that this is true?
There are mutants like DXS mutants that would perturb the production of mycorrhizal, and I mean the splitting products of carotenoids. One is mycorrhizal, and there's this other. Yeah, exactly. Thank you. And you do see a reduction in colonization, but you don't. I mean, maybe one should look at it again, but back in the days when they tried to look at the effect on arbuscular lifespan, there wasn't much to be found. So at present, on the basis of what we do know, there is no effect necessarily directly on the arbuscular lifespan, and so which was what was proposed, that you would, I think you had this brooming type of metaphor, that you need these compounds to actually say, get out now, enough, and so then you would expect a different lifespan if you interfere with the compounds, and they could not nail that. On the other hand, back then we just altogether in the field didn't have these phenotyping possibilities. So maybe one should look at it again.
Okay, thank you very much. Students are waiting for you.
There's a question in the chat box from Jianing Mi. So yeah. I'll read it up. Thank you so much for your excellent talk. I have a question. Do you have any idea about other signaling molecules that can act or play a role in the communication between AM fungi and plants?
Thank you very much, Janine. So yes, I mean, historically, it has been shown that in the pre-symbiotic crosstalk, you could stimulate different fungal growth patterns by different plant-released and fatty acids and isoflavonoids, flavonoids. These efforts have somehow a little bit ceased because there was not too much of encouraging data coming from flavonoid biosynthetic mutants long time ago in the 90s in maize, which were perfectly well colonized. So while these compounds eventually have an effect on fungal growth patterns with regard to branching, it is unclear whether there is, or at least it doesn't look like that they do very much with regard to the ability of the fungus to colonize. Having said that, I'd also like to be precise when we think about strigolactones. What is precisely the effect on the fungus? So when you look at the fungal metabolism, the perception of strigolactones by the fungus, and we don't know how the fungus perceives strigolactone. It doesn't have a strigolactone-like receptor protein. And I mean, there's no gene encoding any related protein in the fungal genome. So we really don't know how the fungus is perceiving strigolactone. But that perception makes or leads the fungus to an absolutely fundamental decision beforehand, before detecting strigolactone. The fungus is growing in sort of in a relatively economical fashion through the soil in search of a host root. It does not burn its reserves, its carbon reserves. So it can, after 2-3 weeks of not finding a host, it can withdraw the cytoplasm back into the spore, right? And so you have reversible germination, unheard of, totally crazy. And it can also germinate again. And when maybe times are better and maybe there is a host root. So that is not happening anymore, cannot be happening anymore once the fungus has perceived strigolactone, because then it burns its reserves. It starts to grow vigorously, branch vigorously. So that is when the fungus already commits to the symbiosis, because it knows the host is very close by. Right. So, but then if you now have strigolactone biosynthetic mutants, all of that is not happening. The fungus doesn't see where the root is. It's slightly disoriented, but it might just accidentally tack onto a root. Boom, there it is. There's a root. Do you need strigolactone for colonization then? Oh, the fungus gets readily into the root, right? So the reduced colonization that we see in strigolactone biosynthetic mutants is due to the fact that the fungus is not navigated to the root. Once it starts to colonize, you come back, 3 weeks later, it's beautifully well colonized, the root. So there are other cues, other signals that the plant administers, I guess, and that are then required for root colonization. What these are, we would love to know.
We'll have one last question from Rod Wing.
Hi, Uta. How are you?
Very good to see you. Wonderful. It's been a while.
Yeah, I enjoyed your seminar.
Thank you.
I just had a simple question. The D14L mutant, does it have a phenotype, in the field for rice? I mean, does rice, is there any penalty at all, or maybe under stress conditions or anything?
It would be hugely interesting to test that, right? I mean, we would love to know that. No, we don't know.
Okay.
Can you send us the mutant? Can you just send us the mutant? We are trying to get it from your lab all the time, but it's so difficult to transfer the material.
Yeah, it's horrific. Absolutely. It would be wonderful to do that, Salim, together. That would be great.
Because we know we have several ideas that we want to test, and we are trying to get it, but it's so complicated,
Yeah, and we're slowly coming out of the fog of the new Brexit regulations. So we have struggled. We've had a number of requests for different materials from different areas, and anywhere we've struggled to send material to. I hope we've now learned the ropes. Can I quickly ask with who did you have contact about that, Salim? Because then I can directly say that.
Juan, can you please answer?
Oh, it was Juan. Yes, I can.
Yes.
Juan
Hello.
Ciao, Juan. Hi.
Yeah, nice to meet you. Yeah, we were in contact with John Ming. Yeah.
Okay. Yeah, because I think we now have learned. Yeah, as I said, we now have learned the ropes and know how to do it. Yeah, let me touch base with her. Sorry for the delay. Really sorry.
Okay, thank you. No problem.
Okay, so thank you very, very much again, Uta, for a lovely seminar and a great discussion. I think when we invite you back in a few years from now, you'll tell us all about how you've engineered the system into Arabidopsis, and we've got Arabidopsis with Mycorrhizal.
Well, the Arabidopsis misses 140 genes. I'm not intending to do that.
Okay. Anyway, thank you very, very much indeed.